Back for part two. So this is where we get to actually use our petri dishes and to sample stuff. So we start with the hand washing experiment. Just ignore the one experiment per table. So this was adopted from the lab manual. We know that hand washing has been given by the CDC as an effective way to reduce, passing germs and infectious pathogens and all that good stuff. And so you can see signs for hand washing literally just about anywhere, especially food kitchens and things like that across America. And so let's say you were an alien and you came down to visit our planet and you're like, okay, why are all these signs out? So this would be an observed phenomena. Now you can ask the question why. So you see the phenomena as all these hand washing signs everywhere. You can ask the question and say, why are these hand washing signs there? And then you could pose a hypothesis, which is that there's some mechanism that hand washing has an effect on. And maybe that's why. And so we can test that. We can say, okay, what's the mechanism? What's our hypothesis? Does or does not hand washing have an effect on the number and diversity of bacteria on the hands? I'll let you guys circle that one. So we're going to test it. So what we want to do first is we want to take one of our petri dishes, and I've got them covered up so that we don't get any dust on them. And I can't tell if this is this, yeah, after and before. So on the bottom of the petri dish, I wrote before and after. I wrote it backwards. So kind of like you're looking in the mirror. That way, when you look from the top, you can see after on the top and you can see before on the bottom. Sorry, my image is mirrored, so it's hard for me to do this. You can see after on the top, before on the bottom. Okay, so we're gonna take our petri dish and you're gonna divide a line. So you're gonna draw on the bottom. You're gonna draw a line down the middle, and one side you're gonna label before, one side you're gonna label after. Now you can write a B, you can write an A, whatever you'd like to do, as long as you can keep the two straight. And so what we're gonna do is we're going to open the plate. Now here's the tricky part. When you open this plate, just remember that you're exposing this plate to dust and mold spores and everything in your environment. Consider this. If you have ceiling fans going, if you have a central air going, if you have anything that's stirring up air around you, turn it off and wait like 10 minutes because that'll allow the dust to settle a little bit. It'll allow the spores to settle so there won't be as much risk of contamination. So you open the petri dish and take your dirty hand and you're going to wipe your finger and cover the entire surface area of before. Okay, so I'm going to do that real quick. I'm even going to tilt my petri dish on its side so I have less of a chance of having mold spores fall on top of it or bacteria spores. Okay, as soon as I'm done, I'm going to put the lid back. That way it's not collecting any dust on it, so nothing's growing. Hopefully nothing's landing on it. Okay, next, wash your hands. Now, usually say you're saying row, row, row your boat gently down the stream. Do that. So go wash your hands, sing row, row, row your boat the whole time, just like you would normally do. And then think about it. Are you going to dry your hands with a used towel, or are you going to dry it with maybe a clean paper towel? So remember, you could also transfer bacteria from the towel to your hands. So I'm going to actually use a paper towel for my kitchen when I go to dry my hands. So I'm going to pause the video. I'm going to wash my hands, and I'll come right back and start it again. Okay, so I just washed my hands. Now, one thing I was careful was I'm going to use my finger to rub it on the plate. And so the finger that I'm going to rub on the plate, I made sure that I didn't turn off the faucet or I didn't grab the paper towel with that finger. I've kept it as isolated as possible. And now we're going to grab our petri dish and we're going to do the same thing. We're going to take our finger and we're going to rub it and try to cover every square inch of the after side. So go ahead and do that real quick. So put the lid down. I'm going to keep my petri dish at an angle so it's not catching as many mold spores, hopefully. Boom. Put the lid back on. Yeah. And now we get to take our parafilm and we get to make a little band to go around the outside of it to seal it in. And so with the parafilm, what you can do is go about 1, 2, 3, go about 3 lengths. Cut those off, and then you're just going to cut a little strip. Make it about 1 inch, maybe 3/4 of an inch. And then we'll take that parafilm and we'll peel it away from the backing. And what this is going to do is this is going to seal the edges around your petri dish. So with the thumb, you're just going to hold it in place and then pull it tight as you wrap it around the outside of your petri dish, forming a seal around it. So you seal up the whole petri dish. And now what we need to do is we need to incubate this for 5 days. So we're just like leave it at room temperature, set it on top of your fridge, right? It's all sealed up. And hopefully we've done a good enough job keeping it clean that we can actually have viable results for our experiment. So that's one petri dish. We got 9 to go. Something we should do though, let's do this while we're sitting here. So if we're doing an experiment, I mean, in this particular case, this hand washing one is definitely like a comparative experiment because, or a comparative investigation because there's no control group. But what I want to do, what I think we should do is I think we should take our petri dishes and I think we should select one of those petri dishes, cut a little strip of parafilm, and then don't even open the petri dish. What I want you to do is I want you to write on the bottom, control. And I want you to think about why we would be doing this. Why would we be taking one of those 10 petri dishes and making a control out of it and not opening it up at all? What would that tell us in 5 days? What could that possibly tell us? So in 5 days, we're going to take a look at these dishes again. Okay. Not even going to open it. I'm just going to seal it with our parafilm all the way around the border, all the way around. Nice and sealed up. No air in or out. And this time, not on the bottom, but on the top, I'm just going to write control. So I just wrote control on that, and that's just going to go off to the side. So at the end of this, I'll tell you why we did that, and it will make perfect sense to you. All right. Prediction. So we can make a prediction. I'm on page 3. We can make a prediction about our hand washing. So we can say, based on my hypothesis and my experiment is going to inform my prediction. So my prediction comes from my experiment. So my prediction will be about the diversity and the number of bacteria that are present on the plate. And that supports my hypothesis or it rejects my hypothesis. So my prediction is the after side of my plate that I just made. Will it have greater or less than the number of bacteria on the before side? So that's just number of colonies. That's not diversity yet. And then I can answer after hand washing, the diversity, that's the different types or kinds of bacteria on the after side. Will the after hand washing side have a greater than or less than when it comes to diversity of bacteria from the before side of the plate? And then we'll cover the results part, how you would graph that next time in the next video. But for now, we're going to move on to our next section. And this one is sampling body parts. So, you ever heard of a 5-second rule? Okay, well, there's an unwritten extension of that rule that if the food is wet that you drop on the floor, you have eating less time before it's contaminated. So if you drop a peanut M&M on the floor or a piece of bologna, you'd probably be fine eating the peanut M&M, but you wouldn't want to eat the bologna. Well, why? Because the bologna picks up all the dirt and gunk off the floor because it's wet. And so for this experiment, you're going to use the same techniques as the hand washing experiment, except this time we're going to use water to help transfer bacteria from our body to the plates. And then you can watch the YouTube video. Go ahead and click that link and watch that video and then come back and join me for this. When we do this, we're going to have to get some sort of clean water. Now, it should not be tap water. It shouldn't come out of your fridge, but I'll show you. So if you have bottled water or you have mineral water, those two things will work because they don't have bacteria in them. Or if you have sparkling water. I actually don't have bottled water, so what I'm going to do is I'm going to grab one of our like LaCroix sparkling waters. Totally sterile. When I crack it, it's going to be pure water. Granted, it'll have some flavors in there, but that's not going to make a difference when it comes to a petri dish. So I'm going to do that right now. Okay, so for me, I'm going to use my Topo Chico. I just opened it and then I put the lid back on so no dust or mold spores fall into the bottle of water. We're going to take 3 more plates. We're going to take 3 more plates. And what we're going to do with these plates is we're going to flip them upside down and we're going to draw a line on the bottom of them. So we're going to cut them all in half. Not like with scissors, but you know. Just going to draw a line through down the middle of all 3 of them. And then what I want you guys to do is pick, you're going to, oh geez, I just leaked paint pen all over that one. You're going to pick 6 body parts. Now, presumably, I mean, you can do this any way you want, really. But what I would do is I would say, okay, pick body parts in pairs that you could sample. So, for instance, I could sample, like I could sample like inside of my ear and inside of my nose, and I put one on one half and one on the other half. So I'm gonna do that. So I'm gonna write backwards. I'm gonna write ear. And nose. Oh, oops. That's going to be one test. And we're going to compare them all together too. So we're not just comparing ear and nose, but on the same plate, it makes sense. Let's do belly button and armpit. So belly button, and armpit. So that'll be my next one. And then finally, let's do hair, like my head. Do hair, and I'm gonna do feet, like foot, bottom. So I'm gonna do foot, the bottom of the foot, and my hair. Okay, so how this is gonna work? Once you've labeled your petri dish, you can flip it right side up, and then you're gonna take a swab. And what I recommend is making sure your hands are clean. So you spray off your hands. Okay. Spray off your hands. Make sure your hands are clean. And then these are sterile swabs that are in your lab kit. So these are actually, you don't have to sterilize these. And then what we're gonna do is we're gonna take this swab. We're gonna dip it into the water, whatever your clean water source is. And then for me, I'm gonna rub it all over my ear. And then I'm gonna take this, and instead of just like streaking across the plate once or twice, I'm going to try to cover every square inch on that ear side of my plate with the Q-tip. Okay, so I'm gonna do that. Take it out, dip it in my water, and then I'm gonna rub it all over my ear, like the back of my ear, the inside of my ear. I mean, hell, if you guys want, you could just like test the I was gonna say you could do the inside of your ear versus the outside of your ear. That might be a fun one to check. So there's a lot of leeway here on how you do that. So I think I've got plenty. Now I'm just gonna pick up my plate. I'm gonna hold the lid above it so no dust gets on it, and then I'm gonna rub it all over every last square inch of this. So I'm gonna hold it, and then I'm just gonna smear it. And I'm twisting the Q-tip, so I'm making sure that the Q-tip comes into contact with every portion of the ear side of that plate. Okay, and I put the lid back on. I can take my Q-tip and chuck it because I'm not going to need it. And then we go on to the next one. All right, so now I'm going to check my nose. So take the Q-tip out, dunk it in the water, and then go all over the nose. So outside of the nose, inside of the nose, and I feel like I'm doing a COVID test. Okay. I might grow some COVID on my plate. No, that won't happen. And I'm going to lift up the plate. I'm just going to rub it and try to cover every bit of surface area on the nose side with my Q-tip. And be very gentle with your plate too. It's super easy to like to tear up the agar, like if you push too hard. And you might find that out the hard way, and that's okay. I mean, we're just learning here. Okay, then we'll take our parafilm, we'll cut another strip. And then immediately we want to seal this up because we don't want any outside bacteria. We don't want any outside spores to fall onto the plate. So my ear and nose is done. Just gonna wrap that around. And I am definitely gonna do all this. I promise you guys that. But I'm not gonna bore you with watching me swab different parts of my body. So do exactly what we just did for that. Make sure that you seal it up and tape it with your parafilm. And then after you're done with that, okay, so you should, after this point, have, we'll skip to inanimate objects. Okay, so we'll so pause your video, go ahead and you guys take care of that. I'll take care of those after I'm done talking to you guys. Now, inanimate objects. So we'll take 2 more plates and we're going to divide them in half. And so these inanimate objects, that one looks like hell. These inanimate objects will be things like your TV remote, your cell phone, you know, stuff like that. So we'll divide the plate in half on the bottom. Okay. And half is going to, we'll pick what we're going to do. I'm going to do cell phone for sure. Cell phone and TV remote. That's going to be one of mine. But you could do anything that's not living. So cell phone, TV remote. My other 2 inanimate objects will be something similar. So I'll say like piano keys. That's a good one. So I'll do piano keys because I got a piano over there. And then I'll do my keyboard, like my my keyboard on my computer. Oops, keyboard. All right, I'm using a paper pen, and I'm tempted to blow on it, but if I blow on it, bacteria from my mouth might end up getting through the cracks onto the plates. Okay, so I've labeled those. I'm not gonna bore you guys with walking around and sampling stuff. You've already seen how I did it before. I'm keeping the lid on my water source. I'm opening up my sterile swab only when I'm ready to do it, and then I'm covering the entire half of that plate. Once you've done your inanimate objects and once you've done your body parts, then you should have 3 petri dishes left. And if you're watching this video, that means that you are enthusiastic about this course and that you want as much points or credit as you possibly can get. So I will give you this. For these 3 plates, okay, if you do these 3 plates, I will give you, and you divide them in half, right? Just like we did these ones. And you can compare anything you want. You can compare stuff outside. I don't even care what you do with them. But as long as you're comparing something and you're using your swabs and, doing it in a studious way, if you do those and split them in half, that means you'll have 6 more things that you can sample. If you do those 6 things and then you graph them out next time we do this, like next week, then I will give you 6 extra credit points on this lab that goes towards your lab grade. So 6 extra credit points for doing those on your own. And that is it. So have fun with this, guys. I will see you for the next video where we actually count the bacteria and make our graphs and stuff like that. So we'll see you later.